Okay, so we're going to go ahead and get started. So my job is to present you with a biologist's eye view of the wonderful mathematical tools that Terrell will be talking about soon. But before, actually, while I begin, I'd like to supplement my PowerPoint of the diversity of life on Earth with this marvelous biology in a box that's been sitting downstairs on the table. Has a variety of different organisms. Tiny little fish, little vertebrate here, a variety of shells, sea stars, and so on. And quite apropos, a phylogenetic tree. So if you're interested, these circulate, and the folks in the office, I'm sure, would be delighted to tell you about them. So if you look around, the stunning diversity of life on the planet is obvious. I mean, you can't walk from point A to point B without seeing a great variety of living things. And people have been trying to make sense of this great diversity for a long time. This is an idiosyncratic and abridged version of the way we've classified living things over time. So we know the ancient Greeks, using their eyes, classified organisms into things that moved and things that were green and didn't move, right? Okay, I mean, it's kind of obvious, right? There are some that move and there are some that stand still, and they're green. So that's the Animalia and the Plantae. And that worked really well until we invented the microscope. And when we invented the microscope, we discovered that there were little green things that moved. Okay? So we needed a new category, right? So that was the genesis of the protista, single-celled or at most colonial things that could be green and could move. Then we got better at microscopy. We invented the electron microscope, which gave us better resolution and allowed us to look inside some of those really tiny things, some of which moved and some of which didn't. And we discovered that they didn't have a nucleus or any of the other prominent organelles that we saw in the other members of their group. So we needed another group. And that other group was called the Monera. That, you know, basically what we think of as bacteria. Okay. And in 1969, Whittaker developed a 5-kingdom model, which took, you know, some of the evidence that had been derived from having good microscopes and also combined it with modes of nutrition. So the Whittaker 5-kingdom model has the Animalia and the Plantae, right? Animals eat other things. Plants feed themselves. It had the Protista, which is kind of a grab bag. Everything that doesn't fit anywhere else goes in there. Those of us who taught microbiology were always very unhappy with that, right? Yeah, very bad classification. There were the fungi, which had cell walls like plants, but were not photosynthetic. Okay? That should be 5. And that worked pretty well. That's probably the model that you all learned, right? No? Okay, so some of you are younger, you know, young enough to have, many of you will have learned the Whittaker 5. Yeah. Okay, so, you know, it had its problems. Nobody liked Protista, but it worked reasonably well for a reasonably long time until we started looking at sequence information. And when we had sequenced enough genes to be able to compare across the diversity of life on Earth, we realized that we really didn't understand at all how this came to be. So using ribosomal sequence information, that is to say, the genes for the small ribosomal RNA. Now, ribosomes are the organelles on which proteins are constructed, right? That's the machinery of protein synthesis. So every living thing has them. And ribosomes have two parts, a small one and a large one. And the small one has one ribosomal RNA. The large one varies a little bit. The bacteria, the very simple, the cells that don't have nuclei, they've got a more simple, large ribosomal subunit than the eukaryote. So focusing on the small ribosomal subunit made sense, right? There was only one gene there to look at. And that revolutionized the way we look at life on the planet, giving us this tree of life. There are 3 domains. The group in blue, called the bacteria, and the group in red, called the archaea, used to be together in Whitaker's Monera, right? So not only were they split apart in this model, but they're designated at a higher level. We're not looking at kingdoms here. This is a domain, and that's a domain. And then all those other interesting things, the plants and the animals, the things we can see, and a bunch of little things that we needed the microscope for, are grouped in the eukaryota because we all have nuclei into which our DNA is inserted. So the ability to look at DNA sequence just completely changed the way we think about life on the planet. We now see that we are more closely related to the archaebacteria than we ever would have suspected, right? It has changed the way that we perceive evolution occurred. We had a very different story of evolution before we knew this. So this is the view now. This is what we think now, right, with partial information. We haven't sequenced every possible thing that there is to sequence. We haven't even isolated every possible organism on the planet. We are still discovering organisms. But we have enough sequence information that it quickly became clear that we needed serious mathematical and computational help. So how do we construct a tree such as this one? Well, first we sequence, right? We can do that. The biologists are very happy sequencing, especially now that we have marvelous machines that do it better than we ever could, 100 base pairs at a time. And then we call for expert assistance from mathematicians and computational scientists. And with that, I'm going to turn the floor over to Terrell.